Hi, thank you guys for coming today. I'd like to welcome you to our Friday seminar. Special guest today is Jeremiah Henning. He's a new professor in the biology department at the University of South Alabama. His research specializes in symbioses, especially mycorrhizae on plants. I'm probably going to talk a little bit about a big monitoring network he's part of and setting up some experimental plots here on Dauphin Island, and I'm hoping to get him teaching in the summer program soon enough. He's a native from Wisconsin. He did his undergraduate and master's at Wisconsin Oshkosh before coming to the University of Tennessee for a Ph.D. Then he went and postdoced at the University of Minnesota, and he started at the University of South Alabama this fall. So with that, Jeremiah, come on up.
Oh yeah, that's a good idea. All right, well, thanks for the introduction, and thank you all for coming out on a rainy day. I'm super pumped to be here. So I've been on the island a whole bunch of times, but I've never been to the Sea Lab, so I'm super psyched to be here today. And so historically I've worked in a lot of montane meadows like this and tall grass prairie. So you're going to have to suffer through listening to me talk about systems like this. But with my transition to south, there's not very many montane meadows or tall grass prairies. And so I'm trying to transition into more coastal sand dune ecosystems. And so that's kind of what I'm going to be talking about today is going to be more in other systems, but it could be similar systems that I'm going to be trying to tackle in the dune systems as well. Oh, I gotta turn that on. There we go. Right, so these are our collaborators. My postdoc advisor, Eric Seabloom and Elizabeth Bohr, who run the Nutrient Network. I'm going to be talking a lot about the work with them today. Grad students and postdocs that I was able to work with at Minnesota, as well as some research I'm going to present from my time at Tennessee and in collaboration with folks at Oak Ridge National Lab. Because I am a new faculty member, I am going to make a shameless plug that I am looking for students to start next fall. So if you know any great students, send them my way. I'd be happy to work with them. So over the last 15, 20 years, there's been a lot of really great breakthroughs with the Human Microbiome Project has allowed lots of technological advances that have really shown the importance of microbes that live inside of us for driving our health, right? So I feel like every day I open up a new paper about some microbe tie to some sort of human disease, right? So from irritable bowel syndrome to Alzheimer's links to cancer links are all somewhat regulated by the microbes that live inside our mouths, our guts, and basically every inch of our body. These technological advances in the human microbiome has allowed us to spill this over into other systems as well. And kind of analogously, we know that plants kind of share the same thing, right? So plants are covered. Every inch of plant space is covered with microbes from the tips of the leaves all the way down into the roots, right? And a lot of the insights have kind of shown us that microbes really regulate the overall health of plants, right? So two groups that I spend way too much of my time thinking about are foliar endophytes. So these can be bacteria or fungi that live inside the root or inside the leaves of plants, right? And so they're really important for protecting plants against pathogens and herbivores by creating all these really nasty secondary metabolites, right? So they create all these metabolites that are pretty similar to LSD. So they deter cows or other herbivores from eating them. They also alter kind of plant response to drought, right? So they kind of regulate stomata opening closures on these plants. So they help plants with water retention in the face of drought. A lot of these fungi and bacteria actually have the ability to produce plant hormones, right? So they can make auxins, or they can make cytokinins, or they can consume them, right? So they can alter the signaling pathways in plants and really manipulate kind of the functioning and the physiology of their plant host. And then they also help with salinity tolerance, right? And so you could think about some applications of their importance for salinity tolerance in kind of dune ecosystems. The other group of fungi that I spend a ton of my time thinking about are fungi that live inside the roots of plants, right? So mycorrhizal fungi live in the roots of almost every single plant, wetland, dryland. Every system has mycorrhizal fungi. These fungi kind of serve some similar functional roles, right? So they protect plants against pathogens. They help plants acquire nutrients like nitrogen and phosphorus. They help plants acquire water from the soil, and in return, these plants receive these fungi receive a ton of carbon from the plant. In some systems, these fungi get about 20% of all the carbon that these plants can fix. So 20% of everything that the plant makes goes straight away to these fungi. And so at the level of an individual, you can see kind of the importance of these fungi for the success of an individual. But what really hooked me into these fungi when I first was starting out is their role at kind of these larger, kind of more ecosystem level scales. Right. So when I was an undergrad, I read this really influential paper by Marcel van der Heijden. And in this paper, he looked at the diversity of fungi within a mesocosm, and then looked at how that shaped the diversity of plants above it, right? And when they did that in this kind of controlled mesocosm study, they found this positive relationship, right? So more diversity of fungi led to could support a more diverse plant community, right? So diversity of fungi begets a diversity of plant community. They also found a relationship with the diversity of fungi and the total amount of plant productivity in that system, right? So more fungi allowed for a higher diversity of plants and a higher kind of production of plants in that system. And this paper was foundation for a lot of studies on mycorrhizal fungi, and it led to this explosion of papers, right? And it hooked me into wanting to study these fungi long term. And so the study by Marcel van der Heijden was really great, but it was this kind of controlled pot experiment. And so I got really interested in thinking about if we could apply this same thing to natural systems. And so one of the first studies that I designed myself was to go out to real systems and to see if this pattern held. And when I did that, I actually found it looked something like this, right? So yeah, there's kind of a positive relationship between fungal diversity and plant diversity, and kind of a relationship between fungal diversity and plant productivity, but it's really, really sloppy, right? It's messy, right? And this messiness comes from the complexity of real ecosystems, right? So real ecosystems aren't just pots with fungi and plants living in them. They're pots with pathogens. There's herbivores. There's climate variation. There's a whole lot of stuff other than fungi and plants kind of interacting with each other. And so trying to disentangle some of this messiness or this complexity has kind of really driven what my research has been working on ever since. Right? So I've spent a lot of time thinking about how different biotic or abiotic kind of filters shape the diversity of plants and fungi, from thinking about how big herbivores kind of shape plant and fungal communities, and thinking about this along kind of long-standing environmental gradients, both locally and globally kind of interactions between plants and their microbes. And I've been thinking about how if we can kind of get a handle on these diversity relationships among the symbionts in their plants, what's going to happen when in a warmer world or in a drier world or a wetter world? So how is global change going to alter these relationships between the plants and their symbionts? And then really the thing I spend a lot of my time is the so what. Who cares if there's changes in diversity of plants and fungi if it doesn't matter, right? Does it matter for the functioning of an individual host, right? So if we change the composition of microbes in a plant, how does that change the function of the host? How does it change the physiology of a host? How does it change the feeding of herbivores in the system? How does it change the amount of carbon storage in that ecosystem? So how do changes in biodiversity affect host function and ecosystem function? Right, and so these are the kind of the three big kind of umbrellas that my research program is kind of tackles. I'm not going to talk about all three of these today, and I'm going to focus in on trying to tease apart how plants and their fungi are responding to global change, and then how hosts and ecosystems are ecosystem function are responding to these global change events. I'm going to just grab a sip of water quick. So one of my favorite global change drivers, I think, is deposition of nutrients. And so I've spent quite a bit of time thinking about how nutrient deposition is altering plants and their symbionts. And so first off, we know when we start applying nutrients to ecosystems, a lot of the work within the nutrient network has kind of shown us that when we add nutrients to an ecosystem, we see a loss of plant diversity, right? So overall plant diversity goes down, but at the same time we see an increase in plant productivity, right? So we lose species, but the species that are remaining get bigger, right? And so we may see a transition from a plant community that looks something like this to a plant community that looks something like this, right? So we lose our diversity, but we are gaining productivity in this system, right? And so within the nutrient network, the nutrient network is a globally distributed experiment that is occurring at about 120 different places around the globe. At each of those 120 places, there's one experimental design or experimental methodology that's applied, right? So at each of these sites, there is a factorial nitrogen, phosphorus, and potassium addition experiment, and that's fully factorial. And then in a subset of the plots, we also set up these large fences to exclude herbivores, right? So it's kind of this experiment, 120 different places around the globe, allows us to really tease apart some of the biotic and abiotic contexts that are kind of shaping ecosystem response to nutrient addition. But one of the things that I really struggled with reading all these papers that have been coming out of the nutrient network is what the heck is happening with the symbionts, right? So what is happening to the fungi that are living in the leaves, living in the roots of these plants in these plots after 10 years of nitrogen deposition? And luckily, when I was finishing on my Ph.D., Eric Seablum and Elizabeth Borer were looking for a postdoc to do exactly that, and so I got to transition into their group and to tackle some of these questions. And so I'm going to first talk about how the foliar fungi are responding to nutrient deposition, and then take a second to look below ground at how the root symbionts are responding to nutrient deposition. And so the first big question we looked at was just, if we go and dump nitrogen, phosphorus, and potassium, how is the diversity of these fungi kind of responding to that? And maybe naively, it's kind of a naive starting question. And we did this at one of the nutrient network sites, right? So we conducted this experiment at Cedar Creek Ecosystem Reserve. So this is maybe my favorite site because I spent a lot of time there. But as a community ecologist, this is the home of all Dave Tillman's kind of classic biodiversity ecosystem function experiments. As the ecosystem ecologist as well, this is home to Lindemann's bog and all the kind of the foundation of ecosystem ecology, all kind of housed in this one field site. It's a pretty cool, pretty special place. But at Cedar Creek, we looked at the foliar fungi in Andropogon gerardii, big blue stem. It's one of those kind of characteristic tall grass prairie species. It's kind of the iconic tall grass prairie grass, right? These things can get 7-8 feet tall. And we looked at it within only Andropogon. We looked at foliar diversity across this nutrient deposition experiment. And when we did that, we just kind of took a snapshot and we found something like this, right? So when we added nitrogen, whether alone or with another nutrient, we saw a reduction in foliar diversity, kind of, right? So it's weak, right? There's a lot of variability in this question, and so we immediately looked at this and we're like, well, there's kind of a pattern, but it's really messy, and we wanted to kind of dig in a little bit further. And then we really wanted to think about, well, these are fungi that live inside of a host that are surrounded by other hosts. And so is it these fungi responding directly to the nutrient deposition, which seems a little far-fetched because these are fungi that only live within the leaf tissues? Or is this a response to nutrient deposition that's kind of mediated through their plant hosts, right? And so we wanted to kind of think about these direct versus indirect effects. Right, so we to kind of walk through kind of a meta model of what we were thinking here, right? So we have our nitrogen, phosphorus, and potassium addition, and we have the outcome of fungal diversity, right? If we had conditions where we had a direct effect, right? So when we added nitrogen, we saw a direct effect on fungal diversity. That could be one potential pathway. We also know that when we add nitrogen, phosphorus, and potassium on these plants, we see an increase in both the standing live and standing dead plant biomass, right? So these fungi are horizontally transmitted between plants, right? So maybe a bigger plant next door to you will have a higher reign of kind of propagules that get to our focal plant tissue. And so maybe there's this increase in propagule pressure on our focal leaf. Additionally, we know that when we add nutrients, we kind of change the composition of the plant community. And we see a change in the overall abundance of the Andropogon plants that are in each of these plots. And if we have some fungi that are more kind of host specific, if we lose Andropogon cover, we may see a loss in diversity in our focal plants. Or if we see more Andropogon in these plots, we may see a higher diversity in these plots if we see more movement of host-specific colonizers getting to our plants. We also know that we lose plant diversity in these plots as well. So maybe there's the loss of plant diversity reduces the overall diversity in the kind of the overall pool of potential colonizers that could get to our focal plant. But these are all kind of independent hypotheses that we could test with a structural equation model, right? And that's exactly what we did. So we constructed these structural equation models. We pulled out different connections to test all these different potential hypotheses, whether it's a direct or indirect effect mediated through different plant pathways. And when we did that, I'm gonna save you a lot of model selection, and I'm just gonna fast forward into the final fit model. Overall, the thing that you should notice is that we found the indirect effect of plant biodiversity loss in our plots led to a loss of foliar fungal diversity in our focal plant species, right? So if you're a focal plant and the fungi that are living inside of you are completely tied to the level of diversity in the plants that are living around you, right? So it's kind of this reduced movement or reduced amount of diversity in the potential pool of fungi around you. And so the next thing after that was kind of thinking about soil fungi. So we know what's happening in the leaf fungi, the foliar fungi. We know that's tied to the plant symbionts. Is that also occurring within the root symbionts as well? And so this is a project that I've been working on with Ilva Leckberg, who's at the MPG Ranch in Montana. And we knew there's a paper from the Nutrient Network that has shown that when we add nutrients to these plots, we actually see shifts in the kind of the guild composition of these different fungi, right? So we see increases in ascomycota, we see some losses in glomeromycota. Just as a kind of a comment, the glomeromycota are a mycorrhizal phyla, right? So we are losing our potential mycorrhizal species. We're gaining other fungal taxa, right? So we're changing phylum composition with nutrient addition, but it's really hard to know anything about what the fungi are actually doing from knowing the phyla, right? So in every phyla, right, so ascomycetes can be mutualists, they can be pathogens, they can be saprotrophs, they can do a lot of different stuff. And so one of the questions that Ilva and I were interested in doing is trying to take more of a functional response. And so what we did was we took the kind of the fungal data from the left paper, and we put that in more of a functional framework, right? Where we organize these different fungal taxa by mutualists, pathogens, or saprotrophs. And the kind of the take-home point that we've been finding is when we add nitrogen, we actually see an increase in pathogens in these systems, right? So higher pathogens when we add nitrogen. And this is across 25 of the Nutrient Network sites. But we actually see the kind of the flip as well. So when when we're adding nitrogen, we also see this loss in our mycorrhizal kind of beneficial fungi, right? So adding nitrogen, we're gaining pathogens, we're losing our beneficial symbionts. And so I guess the next big thing that we're sort of scaling up to is to see if these have any real effects at the kind of the ecosystem level. So does this matter for kind of the longer term function of how these systems are functioning? But we're really not there yet, right? And so, but the next thing that we wanted to do is kind of put it within this SEM framework, right? To think about how changes in the root symbionts, are those also guided by changes in plant diversity, or are they going to be guided by changes in fungal or in plant biomass, or some of these direct effects? And we actually found when we looked at the soil pathogens and the soil AMF, or the mycorrhizae, we saw no connection to the plant community. And it actually seemed that there was a direct effect from the nutrient additions onto the fungi in the soil. So we're actually seeing the same response, right? So we lose diversity in both the leaves and the roots and the soil of these fungal communities, but it's all getting there through different pathways, which is kind of cool. Some are responding directly to the nutrients, some are responding directly are responding through the plant symbionts. And so it kind of makes a little more sense why the soil fungi would be responding to directly to the nutrient addition, right? So these fungi are actually competing with one another to gain access to the nitrogen and phosphorus in the soil, right? And so they can maybe outcompete each other, and we're losing some through changes in that composition or competition dynamic. Right, and so kind of wrap that section up is plant diversity loss and increased plant productivity with nitrogen addition leads to loss of our foliar diversity, our foliar fungal diversity. But our soil fungi are responding directly to nutrient addition. And we're getting to the same overall place. We're losing diversity in both places. How we get there, though, differs amongst these different symbiont compartments, which I thought is pretty cool. All right, so that should be a tease about how these fungi are responding to some global change pressures. And now I'm going to move into, does it matter? Does it really matter if we're losing diversity of some of these symbionts, or are these systems just going to keep moving on? And so the first kind of I'm going to dive into more of a kind of a specific project, transitioning from these globally distributed grassland ecosystems into a more controlled ecosystem in this pot. And so in this experiment, what we did is we took these Populus deltoides. So Populus deltoides is a common biofuel tree. It's cottonwood. Right, so it's used for biofuels in a lot of parts of the country. And what we did is we inoculated these biofuel trees with different bacterial strains. And so we inoculated these strains individually first. Right, so on the tree, and we knew that each of these bacterial strains were having different effects on the host plant. Right, so one of these strains, so this yellow Pseudomonas, when we inoculated that on the tree, we saw these really big leaves produced on the tree. Right, so it increased the leaf production in the plants. When we would inoculate this red Pseudomonas, we would see everything getting allocated to the roots. Right, so we would see these really dynamic, well-branched root systems. Interestingly, these three Pseudomonas strains, if we just did a 16S sequence read on these bacterial strains, would be the same OTU, right? So phylogenetically, very, very, very similar. But because we had fully sequenced genomes of these, we knew that there's some small tweaks to the kind of the genetic machinery that these different Pseudomonas strains had. And then we also included this Burkholderia strain, which was super prevalent in our field studies. When we would look at the different strains colonizing these trees in the field, we would see lots and lots and lots of this Burkholderia strain. And so we wanted to include this Burkholderia strain as well in these communities. And so we mixed these 3-member communities in a foliar factorial way, creating 4 different bacterial communities in combination of these 4 different strains, right? And it looks something like this, right? So we had each community missing one of the members. So we inoculated these trees, we let them grow for about a month, and then we came back and we harvested the roots to look at kind of the final composition of these bacterial communities. And when we did a qPCR on the roots, we found something that the communities looked something like this. And I thought, oh crap, this project sunk, right? Because every time we would inoculate this Burkholderia strain into this community, it immediately took over, right? So 97, 98, 99% of the overall community composition was driven by this dang Burkholderia strain. And so, having spent a lot of time reading some of the classic grime work, thinking about the mass ratio, I immediately started thinking, well, all these strains are going to look, all these communities are going to probably look like the Burkholderia alone. So I'm probably going to have 2 phenotypes that exist from this experiment. One of these 3 plants, and then the non-Burkholderia tree. And I was pretty disappointed. But I kept pushing ahead and decided to keep looking. Right? And so I was interested in thinking about what the overall phenotype of these plants were going to be. And so what I did was I separated biomass, whether the tree was allocating resources to the leaves, towards the stems, or towards the roots, right? So looking at just changes in allocation of resources around the host plant. And when I looked at the first 2 communities, I was really, really disappointed, right? So our non-Burkholderia and our first Burkholderia strain communities looked almost identical, right? So overlap in where they were allocating the resources, and I was pretty bummed. But then when I looked at the other communities, I actually found that there were changes in allocation within these resources, right? So when we had these red Burkholderia strains with our red Pseudomonas strains with Burkholderia, we saw more resources going into above ground portions of the plant, less going into the below ground, right? So, and kind of counter to some of the work of Grime, we saw that the combination of these kind of rare or low abundant strains were driving the overall allocation patterns of biomass. But we also found similar things within leaf tissue chemistry and chlorophyll content of these trees as well. And so even just shifting just closely related microbes could alter where and how these trees were allocating resources. And so that's great for the individual, but how do symbionts kind of scale up to kind of the ecosystem level scale, right? Can we manipulate the symbionts and see meaningful results on carbon storage in our carbon fluxes in these ecosystems? And so now I'm going to kind of zoom back out. So this is going back to Cedar Creek. So this is the Dave Tillman classic biodiversity ecosystem function experiment, right? This is kind of the foundation of the biodiversity ecosystem function relationship, where increasing plant diversity increases the function of an ecosystem. If you work at Cedar Creek, it's always measured as standing plant biomass, which they call plant productivity. But that increases to a point and to some sort of saturation point. And this has been found over and over again across a lot of different studies. There's a lot of arguments about why this happens or what the mechanisms are, and it doesn't matter. I'm just thinking about this kind of biodiversity ecosystem function relationship in general. But within this experiment, about 10 years ago, they also started overlaying other treatments, where they started knocking out different symbionts in this community. Right, so they started every 2 weeks through the summer, they start spraying this field with foliar fungicides to knock out all the foliar endophytes in this community. They also apply soil fungicides to knock out the root and soil fungi. They spray for insecticides all over these plants to knock out insects in a subset of these plots. And then they also have these plots where they just kind of napalm everything and just spray the crap out of these plants with all sorts of things, right? So they remove soil fungi, the insects, and the foliar fungi. And so in collaboration with 2 Ph.D. students in Eric and Elizabeth's group, Mayank Kohli and Megan Wilcots, who are more ecosystem kind of focused, I convinced them and Eric and Elizabeth to start measuring carbon fluxes within these plots, and to think about how diversity and symbiont, knocking out these symbionts, kind of regulate carbon fluxes overall. Right, and so there was an early paper by Eric Seablum that showed when when we spray foliar fungicide, we actually see an increase in standing plant biomass, right? So it seemed that the removal of the fungi actually helped the plants acquire more biomass, which is kind of a weird finding. I guess we just knocked out all the potential pathogens out of this system. And so we were interested in seeing if we could follow this through to the flux levels. And what we did net ecosystem exchange measurements, right? So we went out to a system, we dropped this cube to seal up the system. In the middle of that cube is a CO2 sensor, and we're just measuring the net flux of CO2 moving from the atmosphere to the plants through photosynthesis, and what's getting respired back out of the plants and out from the soil as respiration back to the atmosphere, right? So we're just measuring that kind of the net flux of carbon change. We also can drop shade cloths over the top of this cube while we're taking these measurements to create a light response curve, which allows us to separate net ecosystem respiration from gross primary GPP, gross primary productivity in these systems. So we can really tease apart that ecosystem fluxes in these systems. And maybe as you kind of suspect, when we did this in these systems, we see more biodiverse plant communities have a higher rate of GPP, right? So maybe it's not an unexpected result, but in kind of matching what Seablum's group found initially is when they applied the foliar fungicides, we actually saw a similar increase in GPP, right? So we are removing pathogens that are leading to higher flux rates, so more GPP or more carbon is being fixed per unit of biomass when we remove these foliar fungi. Looking at net ecosystem respiration, we kind of find the same pattern. More plants are respiring more carbon, right? So the more diversity of plants in the system, we're just seeing an increase in the respiration that's coming back out of the system. And we also see that same pattern with the foliar fungicides, right? So foliar fungicides are also leading to higher respiration rates. But we know that those plants also have bigger plants. These plots have bigger plants when we're spraying foliar fungicide, right? So bigger plants can fix more carbon, but if we also are fixing carbon at a faster rate, so that could lead to bigger plants. And so it kind of led to this kind of round robin kind of discussion within our group of which was driving which. And it kind of led us to think about what these flux rates look like per unit of plant biomass. And so trying to account for the changes in plant biomass and thinking about flux rates per unit of plant biomass. And when we looked at it per unit of plant biomass, we actually find very different patterns. So we pretty much lose our effect of biodiversity, right? So more biodiverse plants aren't really leading to higher flux rates, which suggests that the increase in GPP is completely driven by that over-yielding or the higher amount of biomass in those plots, right? But we're also seeing, we're still seeing our response of our foliar fungicide, right? So applying foliar fungicide is still leading to an increase in GPP, and it's leading to more respiration per unit of biomass, right? So the fungi are actually slowing down this system somehow, and maybe it's from pathogenic effects. And these effects are most pronounced in our monoculture plots, right? So this kind of suggests to us that it's pathogen accumulation within our monoculture plots that are really having this big effect on our carbon flux rates in the system. All right, so I'm wrapping up to kind of reiterate some of the points. So in this within our does it matter portion, if we change the composition of our microbiome, right? We saw that just subtle shifts in the identity of the microbes living in a plant could alter where plants were allocating resources, how much chlorophyll they were able to get, how much nutrients they were able to acquire from the soil. And knocking out different symbiont communities or changing symbiont communities will likely alter long-term kind of carbon storage processes in these systems. And with that, I'd be happy to take any questions y'all might have. No, that's one thing we have. That's one thing that we're missing, and so that could be included in our direct effect. Is we might be missing. It could be an indirect effect that we're just not accounting for, and stoichiometry is the obvious one. That's ultimately what we think is driving the soil fungal response, because we know that when plants are changing, when plants gain access to nitrogen or phosphorus, they can disassociate from their symbiotic partners, right? And so that's ultimately probably what's occurring in there. Those data, we're trying to get those data now, so we're going back to some of the older plant samples and trying to pull some of those out. Exploitation, like that they're. Oh yeah. Yeah, so that we're not accounting for that now, but ultimately that's probably absolutely what's happening, especially in the controlled mesocosm, the inoculation experiments. There's not many studies like that are actually including more than just one microbe at a time, or maybe two microbes at a time. And so that's definitely like one of the long-term things about thinking about the microbe-microbe interaction, because we know that some of these strains, that Burkholderia strain, for instance, is only one or two pieces away from being able to fix nitrogen. But if it's grown with the right microbes around it, those microbes can cross-feed it what it needs to be able to actually fix nitrogen in roots. And so there's absolutely some cross-feeding. There's competition among the microbes that we're not accounting for currently. But I think that's ultimately where we need to go. Oh yeah, yep, absolutely. Yeah, absolutely. Yeah, there's nematodes and other things that feed on the fungal hyphae in there, and they also consume the roots themselves. That's also another layer that we're missing that I want to start add in eventually. Oh, absolutely. Yeah, you're absolutely right. The short answer is kind of, but not really. The longer answer is we've done it in some experiments where we've tried to control for soil type. In a lot of these areas where we set up like the Tillman's biodiversity spot, They started from like kind of scorched earth, where they kind of just tilled everything down and kind of tried to homogenize the field as well as they could. There are definitely hotspots of different patches of clay and silts out there, but the overall that site is mostly just a big sand plain anyway. And so overall out there, there's very little carbon content because it's just sand. But yeah, in other experiments, I have done more controlled things for soil type, but in a lot of this stuff, no, I'm not accounting for it. I'm a complete plant microbe person. Yeah, it's definitely something I need to think more about. Absolutely. Yeah. I don't know. That's what the excitement about jumping into the dune systems are. We know that these fungi are super, super important for plant establishment in these dune systems. And so I don't know. Yeah, that's one of the exciting things I'm hoping to find out. So I've done work in some of the Great Lakes dune ecosystems, and we see similar-ish kind of pattern. Their importance is there. The fungi are important, but yeah, we don't know. I guess that's why I'm excited to transition into the dunes. I think it depends on where you go, right? So around Cedar Creek, it's a huge problem, right? Because it's an agricultural hotspot. And so there's a ton of overwash of nutrients that are running through these systems because of the application in ag settings. We also have a ton of industrial deposition that is occurring. Maybe not around Cedar Creek, but a lot of the sites I was working on in Wisconsin, because our proximity to like Chicago and a lot of those kind of industrial areas. And so we see it's probably not as drastic as a lot of like the base of the Mississippi, right? It's definitely not that drastic, because we're the start of the chain. We're starting the problem, and you're just catching all of it.